Hi everyone. Today's reminders are about checking your grades. So each week you want to make sure you check your grades on Canvas. Make sure they're correct. And if you find an error on your grade, show me your graded work so that I can take a picture of it and email it to me so that I can see that and update the grade in Canvas. So today's lecture is on sensory systems. It's chapter 10 in your book. And when thinking about sensory, it's all about afferent input, right? So all these systems are going to be ones that detect stimuli. And each receptor is going to have its own specialty. So we have some stimuli, light, touch, temperature, for different modalities of sensory input. And those are each going to be converted into electrical signals, right? For your brain to perceive something, it has to turn that stimulus, whatever it is, into an electrical signal. And this process is called transduction. That input then goes to the brain to be integrated. And there's different kinds of receptors. Okay, so we talked about some of these in lab already, so you'll review some of these and add on some more. The ones we talked about in lab are photoreceptors, chemoreceptors, nociceptors that sense pain, thermoreceptors, mechanoreceptors, and osmoreceptors. And then we're going to add on one they mentioned in your lab manual, and these are proprioceptors. So proprioceptors are the receptors in muscles that help sense body position and fine motor control. So you know you have these because if you close your eyes, you can still feel where your arms are in space, even though you're not looking at them, right? So you can feel the position of your body. And this is really helpful in coordinating movement and things like sports. Okay, so we talked a lot about the nervous system, and now we got to think about how we're going to code these signals from sensory receptors in the brain. So the first thing to note is that when you see an action potential in a neuron, potentials and sensory receptors are going to look a little bit different. Okay, so we're going to talk about local graded potentials of sensory receptors. These look like the excitatory potentials that you see in neurons. Okay, so here's a receptor. You can see these graded potentials, and as they get larger and larger and larger. Okay. And we talk about two kinds of receptors, those that are phasic and those that are tonic. The phasic receptors are fast adapting. So how this works is that if you apply a stimulus, you get a lot of action potentials or graded potentials early on when the stimulus is applied. And over time, you get less of them until the stimulus is withdrawn. For tonic receptors, though, they're slow adapting, meaning that you apply a stimulus, action potentials occur, and they occur at the same rate until the stimulus is withdrawn. So they're not adapting to the stimulus. The phasic receptor is fast adapting, meaning it's responding less over time. So when we look at this in terms of generator potentials, in tonic receptors, the generator potential produced is proportional to the stimulus, okay? So what I mean by this is that if you have some kind of stimulus, right, maybe pressure on your skin, if you get a little bit of a stimulus, a little bit of pressure, you'll get a small graded potential in the receptor and no action potentials down the neuron. Medium pressure, you get a large enough generator potential that gives you some action potentials. A large stimulus gets you a large generator potential and a lot of action potentials. And your brain can then perceive this information as strength of the stimulus because a lot of action potentials means a strong stimulus, a few means a medium, and none means nothing. Okay, so those phasic ones are the fast adapting ones. The rate of action potentials to the brain quickly decreases as the stimulus is maintained. Example of this kind of receptor is called a corpuscle. These are going to be ones in your skin that alert us to changes in the environment. Okay. Tonic, slow adapting receptors, where the rate of action potentials stay constant, are going to be things like pain receptors and free nerve endings. So these are going to be things that if you have a painful injury, for example, that pain continues. Your body doesn't adapt to the pain and you don't feel it less over time. Okay, so I want you to review all these different kinds of receptors in the skin, these cutaneous receptors. I want you to know the terms and functions here, okay? This will be Merkel's discs, root hair plexus, Pacinian corpuscles, Meissner's corpuscles, the free nerve endings, and these Ruffini endings. All 6 of these in the skin have different placement within the skin and different functions. So this is how your skin can sense so many different kinds of things, right? See how an indentation can be sensed by a Merkel's disc compared to a light brushing sensed by the hair follicles. So some interesting things about cutaneous or skin sensations. There are far more receptors for cold than there are for warm. The cold ones are on the surface and the warm ones are deeper. And if you experience something really hot or really cold, that's interpreted in your brain as pain. So something doesn't have to stimulate the nociceptors to be sensed as painful, because really hot and really cold things are sensed as pain in your brain. And capsaicin and chili pepper is perceived in your brain as heat and pain. Peppermint, menthol, is both is going to be perceived as cold. And itching is caused by inflammation and histamines. There isn't an itch receptor. And itch is actually a really interesting phenomenon in medicine and a really common problematic symptom because there isn't an itch receptor. There is histamine and different compounds that make you feel itchy, but it's not one receptor that you might be able to block. So if someone has a symptom of itch as part of their injury or recovery, it can be really difficult to treat. Okay, so some things I want you to connect from lab to this lecture, and I would like you to remember from lab, are going to be the neural pathways involved. We talked about referred pain in lab, receptive fields, and 2-point touch threshold. Okay, so those are going to be things to review from your lab and read more about if you've forgotten them. For taste, sometimes called gustation, taste buds are these specialized receptors that contain 50 to 100 specialized epithelial cells that have long microvilli. Okay, so here's a taste bud, and you can see that there's little taste hairs on the top, and then the cells are inside, and there's supporting cells kind of underneath here until it all goes onto a sensory nerve fiber. Each bud has the 5 types of taste cells. Okay, so it used to be thought that the different tastes were tasted on different parts of the tongue, different regions, but we now know that each taste bud can taste the 5 main things we can taste. So it's not about regions of the tongue. Okay, so what are those 5 tastes? The taste cells are actually specialized sensory epithelial cells that behave like neurons. Okay, so they send neural signals back to the brain, but they are actually sensory epithelial cells. Okay, so how this works. When you're tasting something salty, it's because sodium is depolarizing the cell. When you're tasting sour, it's going to be hydrogen ion depolarizing the cell. Sweet is sugar, and so it's causing depolarization of the cell. And umami, a savory flavor like you find in meats, are amino acids glutamate and aspartate depolarizing the cell. And bitter, it's going to be quinine or toxins depolarizing the cell. So essentially, these cells, by being depolarized by different compounds, then can go can synapse onto your brain, and that will eventually let you taste that flavor and perceive that flavor. So after the cell is depolarized and fires the action potential, it releases neurotransmitters just like any other neuron, right? So, and those are going to then go to your brain, synapse on your brain, and tell you whether it's a flavor that you like or don't like, right? Maybe you want to eat more or less of that thing. And the neurotransmitters then stimulate these sensory neurons, which signal the brain. So you have this whole pathway from the tongue back to the brain that's going to perceive these 5 flavors. Here's what they look like. We said salty is perceived when sodium depolarizes these cells. So here's sodium coming in, causes depolarization. Remember, neurotransmitter release is a function of calcium channel opening. So as this action potential comes down this down this cell here, you have the opening of calcium channels. Calcium influx into the cell causes neurotransmitter vesicles to fuse with that membrane. Remember that docking mechanism from the nervous system chapter? And when neurotransmitter is released, then that's stimulating the sensory neuron that will eventually synapse back on the brain. Okay, so sour, same thing, but just going to be hydrogen ion. So hydrogen ion causes depolarization. That depolarization causes opening of calcium channels. Calcium comes into the cell. Neurotransmitter vesicles bind. Okay, so that's how it works for salty and sour. We need G proteins for sweet, umami, and bitter flavors. These are called gustusins. Okay, so for sweet and umami, if it's sweet, it's sugars. If it's umami flavor, it's amino acids. When one of these things binds to the taste bud, a G protein is going to activate a second messenger that closes potassium channels. When potassium channels close, you have a slight depolarization and neurotransmitter released. Okay. And for bitter, it's a little bit different mechanism where that quinine is binding here, activating our G protein, second messenger activated, and now you have calcium not coming in from the outside, but released from endoplasmic reticulum stores. Okay, so there's not a lot of free-floating calcium in your cells in general. There's a lot of calcium in the extracellular fluid, and any internal calcium is stored within the ER. But either way, what happens here is second messenger causes an increase in calcium inside the cell, but it's coming from the ER. And we said calcium is involved in that mechanism that causes the neurotransmitter vesicles to dock and bind and release neurotransmitter then into that synapse. Okay, so now a little quiz. Which of the following cranial nerves conveys gustatory information? I'll let you think about that for a second. And if you answer D, that's correct. Both the facial and the glossopharyngeal are correct. Where on the cerebral cortex is taste information sent? I'll let you think about that one and look it up. Okay, let's talk a little bit about smell. So gustation is one of our chemical senses. Smell is the other chemical sense. And in olfaction, each bipolar sensory neuron has one dendrite that projects into the nasal cavity, where it terminates in a knob containing cilia. The cilia and plasma membrane have the receptor proteins for odorant molecules. Okay, so move this down a little bit here. Here's the nasal cavity. Here's those olfactory neurons that have cilia on them. And then it's this bipolar sensory neuron, meaning it has one projection coming down here and looks like a second one coming up here. So here's a good example of different shapes of neurons, right? They look different. Okay, so here they are, olfactory receptor neurons synapsing back in these glomeruli here, and then on interneurons, and finally go back to the brain. You'll notice too that these olfactory receptor neurons have to go through what's called the cribriform plate of the ethmoid bones. You might remember this bone from anatomy, which it has little spaces there for those neurons to go through. Okay. So each neuron or receptor makes specific protein receptors for specific odorant molecules. So there's approximately 380 different receptors. With a combination, we can smell about 10,000 odors. Binding of the molecule causes a second messenger reaction with the G protein. And there's adenylate cyclase involved. So you know that whole pathway, right? You know that whole pathway of the G protein and adenylate cyclase. So go back and review that if you've forgotten that one too. When this happens, the cell is depolarized and it fires an action potential. And this sounds pretty amazing, right? 380 receptors, 10,000 odors, but compared to someone like a dog, we are not the best smeller in the animal kingdom, right? So we have a decent nose, but compared to other organisms, we might not be as good at smell as some other animals. Okay, so once that cell is depolarized and fires action potential, a synapse with a second order neuron in the olfactory bulb and then the brain. So we get a couple steps here: G protein coupled receptor, action potential, synapse, a second order neuron in the olfactory bulb, and then to the brain. And there's a direct connection from the olfactory bulb to the cerebral cortex, which makes connections with memory regions of the amygdala and hippocampus. And you might remember the amygdala and hippocampus especially for things like spatial memories, and the amygdala for emotion, right? So whenever we connect emotion, we use the amygdala and connect to a sense, that sense becomes very strongly emotional for us. It can be a fearful emotion or a happy emotion, sad emotion. But when you connect it to that, the memory is retained longer and increases. So this direct connection makes these smell and memory a really strong connection. All the other senses pass first through something called the thalamus before going to the cortex. Okay, so smell, we get a direct connection to the cerebral cortex from the olfactory bulb. Other senses synapse through the thalamus first before going to the cortex. So what we get then is that certain odors can evoke really strong or emotionally charged memories. I think it's meant to say odors, certain odors. So certain smells can make us evoke really strong or emotional memories. And can different odors promote particular moods or memories? And probably you know the answer to this is yes, right? Is that if you think about, maybe it's the smell of someone's cooking that you grew up with, right? Maybe it's a certain perfume or cologne that you remember smelling. So they can definitely connect to a lot of these things. So the next one we're going to talk about is the vestibular apparatus and hearing. And all of these structures in the ear you probably remember from anatomy, but we'll review some of them here. So the external part of the ear, the outer ear, consists of this auricle here, and then the external auditory meatus, terminating in this tympanic membrane here. And those tiny little ear bones, the auditory ossicles, go from that tympanic membrane to this tympanic cavity here, okay? And the cochlea is in there too, right? And then you have this part that's middle ear and considered inner ear. So remember, there's also the cochlea, which has the hair cells inside of it, and also the semicircular canals, okay? So the vestibular system and the hearing system are all part of this ear apparatus. Okay. And so the inner ear has to do with orientation with respect to gravity, okay? So here we see these canals, right? And even anterior, posterior, and a lateral canal with this utricle and saccule inside. And this is going to move and be sensed inside such that it goes back this vestibular nerve to give you that orientation information. Okay, so in the inner ear, there's these hair cells. Okay, and so what it looks like when it's at rest, these hair cells point straight up. Okay, and so there's a kinocilium next to a couple of stereocilia, all on this cell membrane. And if the membrane is pushed down, it's depressed, these are going to bend, and that acts as stimulation. Okay, so at rest, the hair cells are just kind of bopping along, action potential, normal. When something happens, it's stimulated, it gets way more action potentials. Okay, if they bend the other way, such that it pulls out the membrane, that inhibits this such that you get a decreased action potential. Okay. So these semicircular canals contain endolymph with a high potassium level, and the fluid moves according to body position and bends those hair cells. So if the stereocilia bend towards that kinocilium, you get a potassium influx to depolarize. If stereocilia bend away, they hyperpolarize. So these hair cells are therefore mechanoreceptors, right? Because the movement of them is what's giving you the perception of position in space. This signal goes to the vestibulocochlear nerve, that's cranial nerve 8. And the cupula bends in the opposite direction of the head movement, okay? So they lag behind the endolymph, and again, kind of give you this rotational information. Okay. So these otolith organs, the utricle and saccule, also have hair cells that bend. The otoliths are actually calcium carbonate crystals, and the utricle is sensitive to horizontal acceleration. So you can actually feel a lot of different things, right? If you close your eyes, you can feel whether you're spinning, whether you're moving, all those different kinds of movements, right? And so what happens here is when your head is upright, you get the hair cells and supporting cells are kind of perpendicular to each other with the otolith sitting on top. If you bend your head forward like this, now we have the stereocilia of the hair cell band stimulating neurons. The otolith membrane sags because these otoliths literally fall to the bottom of this too, and that makes you feel like you're moving your head forward. So utricle is sensitive to horizontal acceleration. The saccule is sensitive to vertical acceleration. Now, moving on to the hearing part of this apparatus, the effective sounds are different for different frequencies on this basilar membrane. Okay, so here's the tympanic membrane of the ear. Here's those three auditory ossicles, the malleus, the incus, and the stapes. And then there's the oval window here. And so if we look at three different frequencies of sound, right, 500 hertz, 2000 hertz, 20,000 hertz, we see that this is going to affect this basilar membrane differently. Okay, so just like the semicircular canal, the cochlea is also full of endolymph and hair cells with stereocilia. And so the sound is a vibration, right? And it moves the fluid, which bends the hairs. So an action potential is then created. A louder sound makes more action potentials. Low-pitched sounds stimulate hairs at the far distal end, while high-pitched sounds stimulate the proximal end. And so it looks like this. When you look at the wavelengths of sounds, a quieter sound has a lower amplitude. A louder perceived sound has a higher amplitude, a larger amplitude. Lower pitch is going to look like this, big waves. Higher pitch is going to be a lot faster waves. And so we look at these things in different ways. We measure this, we call this the period. So between two of the peaks here of a low frequency wave versus a high frequency wave. So you'll notice the period is different. Low frequency wave have longer periods. Higher frequency waves have shorter periods. And we just said amplitude, low amplitude gives you a low frequency sound. High amplitude is going to be sound higher pitched. Okay, so that's very briefly about hearing. And now I'm going to talk about the eyes and sight. So remember some of your anatomy, right? You have the internal anatomy of the eye. And we see light passing through this anterior chamber here, passing through the pupil, passing through the lens. The lens is going to concentrate that light then on the retina. So all your photoreceptors are back here at the back of the eye. Okay, the fovea centralis is that point where you have the highest density of photoreceptors. And that information then is going to go back to your brain through the optic nerve. So the retina is actually covered in lots of blood vessels. When you look with an ophthalmoscope, you can see all these blood vessels, okay, with that fovea centralis surrounded by the macula lutea there too. And what kind of is really weird about this is that the image that you see is actually inverted upside down on the retina. So when you have an image here and light moves through the pupil and through the lens, that lens inverts it onto the retina. So the retina initially perceives that image as upside down. Okay. And the pupil is what's going to change to let light in or out. Okay, so in normal light, you're going to have a pupil of a typical size. In really bright light, that pupil is going to constrict, okay, to make the circular muscles of this iris contract. It's going to be a parasympathetic response. So small pupils, parasympathetic response, also happens in bright light. Dim light, or getting dilated at the eye doctor. In dim light, the pupil is going to dilate as the radial muscles of the iris contract. This is a sympathetic response. This is why people tend to look wide-eyed when they're stressed out, is that you're going to have a pupil dilation as a sympathetic response. Okay. And the lens's job is to change in shape to permit accommodation. So you're able to see things at different distances because your lens is going to change. So the lens is suspended there in the middle by these suspensory ligaments. When those ligaments are taut, the lens is thin and you focus for distance vision. When those suspensory ligaments are relaxed and you're instead contracting the ciliary muscle fibers, now you have a nice thick lens and that helps you focus up close. So you might have known somebody or be somebody that can only see well at close distances or far distances. And depending on your age, that might have to do with genetics and just how your eyes are. Or if it's age, it might have to do with a relaxation of these suspensory ligaments so that they can't relax properly, and you end up with an inability to see things up close. So accommodation is really important in the eye. We need to be able to see things up close and far away. So to see distance, that ciliary muscle relaxes. The ligaments tighten, and the lens is flattened and slightly convex. To see close, the opposite happens. Ciliary muscle contracts, ligaments relax, and the lens is more convex. So how do we correct for some of these problems? This is going to be a whole lot about ophthalmology, right? Is that if you have normal vision, called emmetropia, the rays are going to focus on the retina here, and you have no correction necessary. In myopia, though, nearsightedness, what happens is that based on the shape of the eye, the rays are going to focus in front of the retina. So you have to have a concave lens that corrects this. In hyperopia, farsightedness, the opposite's the case. The rays are going to focus behind the retina. And so you might put a convex lens on to correct that farsightedness. Astigmatism is going to be a bend somewhere at the front of the eye. And so the rays do not focus. And so what you need is an uneven lens to correct astigmatism. So you can have these lenses as contact lenses or as glasses that will help correct for these different problems. So this is to review what we just talked about with the vision problems, myopia being nearsightedness. And when the eyeball is too long, you can fix this with concave glasses. In hyperopia, farsightedness, the eyeball is too short, fix with convex glasses. Astigmatism is uneven lenses, so you can fix it with uneven glasses. And one we just talked about is presbyopia, so loss of flexibility in the lens. And this is why, as people age, they tend to need reading glasses, because you get loss of flexibility in the lens, so you can't focus on things that are up close as much anymore. So the photoreceptors in the eye are what's going to actually detect light. And we have two major types of photoreceptors. You have cones that detect color and rods that detect light or dark. Okay? And there's a purple pigment in a photoreceptor called rhodopsin. And what happens in light is that rhodopsin dissociates into retinine from vitamin A and a protein called opsin. This is a bleaching reaction. And so there's a dark adaptation with a max at 20 minutes to make more rhodopsins. This is why if you your eyes are just in the dark and you turn a light on and turn that light back off, you can no longer see in the dark for a little while, right? So we need this time for rhodopsin to build up for a dark adaptation. And how the eye is set up is kind of weird. This whole thing here is the retina. The light, when it comes into your eye, actually has to go through all these different kinds of neurons until it first stimulates the rods and cones. Okay, so it goes through a bunch of layers of neurons, hits the rods and cones. The rods and cones then synapse onto these horizontal cells here, okay, and bipolar cells. Those synapse onto ganglion cells that are supported by amacrine cells. And then those ganglion cells synapse and collect onto these fibers of the optic nerve and become that optic nerve that goes back to the brain. So light has to come through all these layers, stimulate the rods and cones, and then go back. The signal then goes back to the brain through the optic nerve. Okay. We have three types of cones in our eye. One kind of rod. Remember, the rod is going to be a light or dark sensing photoreceptor. The three types of cones we have are optimized for kind of a blue wavelength, a green wavelength, and a red wavelength. And just those three different kinds of cones and the combination of those gives us all the different colors that we can see. Okay. So this is called trichromatic color vision because we have a short wavelength cone, a blue one, a medium wavelength cone, that's a green one, and a long wavelength cone, that's the red one. And the overlap shows, though, that light can stimulate more than one cone. And here's how you get the different colors. So we don't just see in blue, green, and red. We see in all the colors that we can see, right? Pink and orange and everything else, because light frequencies can stimulate more than one cone and more than one type of cone. So during the day, the light bleaches out the rods, so the cones are used. So in the light, we're not actually using rods, we're only using cones. At night, then, is when you're going to be using the rods. Okay. So again, here's just showing it this in a little bit different way. Here's the cones in the fovea, rods in the fovea. And a major difference here, too, is that the rods have what's called convergence on bipolar cells, whereas the cones have no convergence. So it's one cone to one bipolar cell. You might get multiple rods synapsing on a single bipolar cell. And this helps maintain the differentiation we see in the color vision in the eye. Okay. So because of this convergence of bipolar cells, rods have maximum sensitivity to low levels of light. And it's this 1-to-1 ratio at cones that gives them higher visual acuity but lower sensitivity to light. So it's a trade-off between acuity and sensitivity. And luckily we have both, so we can do both things, right? We can have that high visual acuity during the day with that 1-to-1 ratio, and we can use the rods to be more sensitive to low levels of light. Remember that fovea centralis is the part where you have the highest density of photoreceptors, and it's best for visual acuity because the bipolar cells are parted away in that area too. And signals from the photoreceptors are going to meet at the optic nerve and then send that signal back to the brain. There is a spot though in your eye where the optic nerve goes back to your brain where there's no photoreceptors. This is actually called a blind spot, because if you are able to find it, you actually cannot see in a spot. There's no photoreceptors there. We don't normally see that in our vision because the overlap of our visual fields means that your brain fills in that information. So you don't see a blind spot, but it is there in that spot where there's no photoreceptors, where the optic nerve enters the eye. So that's it for today. I'll see you all next time.